here last time. We talked about proto-oncogenes and oncogenes once they become mutated, basically. So now we're going to, do I need a refresher on anything? Let me start with that. Okay. So now we're going to continue talking about tumor suppressor genes. So of course, for the oncogenes, it's kind of like their persistent activation is what causes cancer. But in the case of tumor suppressor genes, it's their inactivation that results in the continued growth characteristic of neoplasms, okay? So tumor suppressor genes are normally present, and their function is to transcribe proteins that regulate growth and stop growth when it's not necessary, and stops the cell cycle when there's DNA damage. So that's what they normally do. So of course, when they're inactivated, they cannot serve their function. The most common tumor suppressor gene that you'll hear about is P53. So this is in kind of an example comparing normal versus abnormal, for cells with normal P53 and cells with mutation or loss of P53. So you have a carcinogen, or something that results in cancer, and when your P53 is normal, it kind of stops that process. So you have DNA damage, which causes your P53 to become activated, and then it causes upregulation of genes that will kind of fix that DNA damage or cause the cell to die. So that's what its purpose is. I'm so sorry. So some of the genes, some of the common genes, there's more of course, but some of the common genes that it results in transcription of include P21, which is a cyclin-dependent kinase inhibitor. And cyclin-dependent kinases are enzymes that cause the cell cycle to continue. So when this is activated, it causes the cell cycle to basically stop, or G1 arrest, and allows DNA repair to take place. GAD45 is an example of a DNA repair gene. Okay, so of course it repairs the DNA. So if the DNA repair, oh, thank you. So if the DNA repair is successful, then the cell is allowed to continue through the cell cycle, and, all is well. If the DNA repair is not successful, then the apoptotic pathway is initiated and the cell is destined to die. Okay? Is everybody clear on that basic pathway? Yes? Okay. So basically, when you don't have P53 functioning properly, none of this occurs. So there's no cell cycle arrest, no DNA repair, and the mutation is allowed to continue through the cell cycle and divide and become part of that genome, basically, or DNA. Okay. So genes that regulate apoptosis. So similar to if you destroy the mechanism to kind of stop the cell cycle and allow DNA repair, if the genes that regulate apoptosis are damaged, then apoptosis cannot occur when it needs to. BCL2 is the prototypical family of anti-apoptotic genes, and there's multiple apoptotic genes, such as DAX. You may have heard of DAX, BIM, VAC, all those. So the tumor suppressor genes and the proto-oncogenes indirectly regulate apoptosis by regulating the transcription of the apoptotic genes. So the apoptosis can be destroyed if you have damage to the apoptotic genes. Apoptosis won't occur if you have damage to the tumor suppressor genes, because remember, all of that stimulates that pathway. And also when you have expression of oncogenes, they also just allow the cell to continue. So all of this indirectly prevents apoptosis. And then you have your DNA repair genes. So remember, the 4 main genes that regulate neoplasia typically are your oncogenes, tumor suppressor genes, apoptotic genes, and finally your DNA repair genes. So DNA repair genes are present to fix the DNA when it's damaged. So if they're not working, you can't fix the DNA. And as you saw in the diagram, again, this and this indirectly are responsible for your DNA repair. Okay, so I don't have to go back to there. I don't think. Okay, so I think you get the idea, right? So it all kind of ties in and affects one another. So yeah, that's kind of it. So some common examples, which you've probably heard of, are GAD45, but particularly the 2 I'm thinking of you've probably heard of are BRCA1 and BRCA2, which with mutations in these genes, they can predispose someone to breast cancer. These are common genes that they look for. Okay? So GAD45, BRCA1, BRCA2, MLH1, and MSH2. Just some common names you may see floating around and to know their DNA repair genes. Any questions so far? Yes.
So if a woman doesn't have BRCA1, then she might get breast cancer, or she does?
Or if it's mutated, it's usually lost. Anything that prevents it from working, whether it's absent or mutated. Usually it's a mutation in these genes that are really predispose someone to breast cancer. These are the genes they look for to determine like your likelihood of developing breast cancer. Where some women get they'll get the DNA testing and they get a mastectomy if they're shown to have this mutated. Okay. I think I read Angelina Jolie. Did Angelina Jolie do that?
She preemptively got a mastectomy because she had the predisposition to it.
Yeah, that's right. And you're not guaranteed to get cancer, if you have these genetic abnormalities, but your chances are increased. So some people want to just take the extra measures. So just some kind of summing it up. So genetic alterations initiate cell transformation toward the cancer phenotype. Development of cancer by affected cells occurs in response to the aberrant expression of genes. And the aberrant expression of genes leads to the aberrant expression of the gene product. Just remember, all genes eventually, as they get transcribed, they result in a protein byproduct. So that whole pathway goes in line. So normal gene product equals a normal protein production. And sometimes you can even check for the aberrant protein. I'm trying to think of an example. Like for example, when I did my PhD, we didn't look at the gene per se, but we looked at the nitrated form of like tyrosine hydroxylase, which is a protein. So that's an example of how you can actually look at the protein to determine if there's a genetic alteration. But that's more molecular and kind of beyond the scope of what I'm teaching. Nothing to know here other than this is just one example of some of the cell circuitry in every cell of the body. Of course, it varies based on the cell and what it does. But these are like all the things that can go wrong. That's kind of the point. Yeah. There's a lot that can happen. And if any mutation in any of these pathways that is not repaired and allowed to go on into the divided cells doesn't have to. You have lots of mutations and nothing ever happens, but just an example of what can happen and the different pathways that are available to. It just reminds me of the complexity of us. Okay, so now we're going to talk about broad categories of things that cause cancer. So there are chemical carcinogens such as smoking and pesticides. There's radiant energy, the one we talked about, that's a complete carcinogen, so a nuclear bomb, massive radiation. And there are oncogenic viruses. Onco, meaning tumor, viruses that can cause tumors. I think we went through this one once before, but we'll go through it again. So when you acquire something that damages the DNA, you can have successful DNA repair and the cell goes back to normal, or you can have failure of DNA repair, particularly through, like we talked about, DNA alterations in DNA repair genes. And that can lead to mutations in the genome of the somatic cells, which are then passed on as the cell divides. That you can have activation of oncogenes and activation of tumor suppressor genes. So everything we just talked about, alteration of genes that regulate apoptosis, or alterations of genes that that cause DNA repair. So with this, you get unregulated cell proliferation, decreased apoptosis, and then you get clonal expansion of these cells that have this deranged gene expressions, and you get a tumor. And so this would be, remember we talked about the 3 main steps of malignancy? What would this part be? It was IPP. So the one with I, initiation, all of this, promotion and progression, when it becomes malignant neoplasm. Okay. So chemical carcinogens are agents that cause genetic damage to cells, which promote neoplastic transformation. You can have synthetic or biological carcinogens. And you have direct acting carcinogens and pro-carcinogens, which require activation. All chemical carcinogens are highly reactive electrophiles that bind to nucleophilic residues in DNA to form DNA adducts. So you have the chemical that binds to the DNA, and that's called an adduct. Most chemical carcinogens are mutagens, but not all mutagens are carcinogens. So that means most chemicals that are carcinogenic cause a mutation of the DNA. But everything that causes a mutation of the DNA does not result in cancer. Kind of more the same, just specifically talking about carcinogens. This is the one I showed. So you have this electrophilic intermediate, which binds to DNA. DNA repair is fine, if not. You have permanent DNA lesion, so this is an initiated cell. Then you have cell proliferation, also differentiation, pre-neoplastic clone. This is promotion up to this point, and then progression to malignant neoplasm. So a lot of repetition on what we've already said. So radiant energy is a complete carcinogen. It's both the initiator and the promoter. It directly damages DNA. And you also have indirect damage to cells and DNA and every other component of the cell from the production of reactive oxygen species. Okay. So these are some examples of radiation-associated cancer. This is more applied to humans, but of course it can be applied to animals as well. I'm not going to read through those. Basal cell tumor is one of the most common skin tumors in the cat. That's why. All right, so lastly, we're going to just briefly talk about viruses and how they result in cancer. So you have DNA viruses and RNA viruses, and they both can cause neoplasm. I'm not going to ask you questions about this, but it's just more for your knowledge, okay? So transforming DNA viruses form stable associations with the whole cell genome, so they actually incorporate themselves in the DNA. Productive infections is when the infection is productive or it allows more virions to be produced. That type of cell is called a permissive cell. And if you see the word non-permissive cell, that means the cell becomes infected but it's non-productive, meaning it does not allow the transcription of more virus protein and more virions, okay? These are the cells where you're more likely to get transformation to tumor cells than non-permissive. Usually with permissive cells, they're going to end up dying. So they allow them to replicate and the cell dies. But the virus continues. Transformation takes place in 2 stages. There's an early phase and a late phase. So in the early function of DNA virus is to prime the cell for viral production, and it's involved in the viral DNA replication. So you have entry of virus into the cell, removal of the viral envelope, the viral DNA incorporates into the genome, and then you can get synthesis of the viral-coded proteins. And in the late function, the genes code for viral capsid proteins, and you get production of infectious virions, which are suppressed in cell transformation. So this is the one in the non-permissive cells where you're likely to get tumor formation. And you don't have to get tumor formation, but the incorporation of that DNA can cause a genetic alteration or mutation, which results in a non-lethal DNA repair, just like everything else, and then you get transformation in the empathic progression. Everybody clear? Because I know I'm kind of going fast. Okay, so here are some examples of DNA oncogenic viruses. You have papovaviruses, which is like the Papoviridae family, which includes polyomavirus. Simian virus 40 is a type of polyomavirus. You hear about that with non-human primates. And papillomavirus. Papillomavirus is very common in our domestic species. Adenoviruses can cause some tumors. I really need to take that out because not so much in veterinary medicine, but more so in human medicine. And this is one of those vectors they use in lab animal medicine, which can cause tumors, and herpes viruses. Some examples of herpes viruses are Marek's disease. I know what Marek's disease is. What animal does it affect? Yeah, birds, chickens primarily. What's the lesion with that? Like what type of tumor does this cause? Lymphoma. So it's a lymphoproliferative. There's been debates as to whether it's just a lymphoproliferative disorder or is it really neoplasia. So there's debates all of them. Yeah, we don't want to go into that. It's really not clear, but it does cause tumors. So, one of the common places you look for that is the nerves. So the sciatic nerve being one of the bigger nerves in the body. And there's a lot of, well, I'll just tell you. And there's characteristic clinical presentations of this. One of is I can't talk this morning. One is affected nerves, thickening of the nerves. Also, you can get iriditis or iridocyclitis, where you have inflammation of the iris. So you may see an eye that's the pupil is constricted and it looks kind of light blue. And also, you can get a folliculitis. So you'll have like a lumpy bump. Like if you take the feathers away, the chicken will look lumpy bumpy because you have proliferation of the lymphocytes in the follicular epithelium. Okay. And the follicular epithelium is actually where replication of this virus occurs. And other birds get it from inhaling the feather dander that is released from these feather follicles. So it's pretty cool. Another herpes reticulum cell sarcoma in monkeys. So if you have viral inclusions with the DNA viruses, where do you expect to see them? The nucleus. And that kind of makes sense, right? DNA. An exception to the rule, which DNA virus will have a cytoplasmic inclusion. think I heard it, pox virus, in which RNA virus will have both intranuclear and intracytoplasmic inclusions. And what type of virus is distemper? Paramyxoviridae, but what's the genus of the virus? Morbilli virus. Okay, so those are kind of your general exceptions to the rule. Very good. Okay, RNA oncogenic viruses are also known as retrovirus or viruses or oncornaviruses. And so if you see that, it means the exact same thing. Okay. Oncorna came from onco RNA, so oncogenic RNA virus. So these contain RNA-dependent DNA polymerase or reverse transcriptase to allow the RNA virus to incorporate itself into the DNA genome. So it uses the viral RNA genome to make a DNA copy of the cell, and then it goes into the DNA just like the DNA virus. So your common coronaviruses include feline leukemia virus, Rouse sarcoma virus, mouse mammary tumor virus, and this is pronounced Yaxietti virus, type D. Yaxietti virus, which is a type D retrovirus. Okay. This one, have y'all heard of this?
Yeah,
Okay.
totally.
So what animal does it affect?
Sheep, right? Sheep or goats?
Sheep.
Yes.
And what lesion does